No further ado, ladies and gentlemen, it's my absolute pleasure to introduce a good friend and a remarkable researcher, Dr. Mihaela Serpe, a true visionary in the realm of brain research. Dr. Serpe is a senior investigator at the section on cellular communication at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. Part of the National Institutes of Health, Dr. Serpe leads a pioneering research lab focused on understanding the mechanism of synapse development and homeostasis.
Thank you,
Dr.
Theo.
Serpe, are you good, wonderful.
Yes, I'm here, and thank you so much for the invitation. I'm going to try to share my screen now. Let me try to do that, just that. Okay, so how is this one?
Yes, we're seeing it. Oh, wonderful.
My screen?
Yes.
Okay, great. Well, again, thank you, Theo and Ramona. What you're doing is absolutely spectacular. Bringing all of us together. You know, most of us tend to be a little lonely in our, ivory towers doing our work. And I'm guilty as such. I'm a dreamer, for full disclosure, and I rely on people like you to get to know this community and to be part of it. So today I'm going to share a little bit of the journey from dreaming to be a scientist to becoming one. It is my journey. I grew up in Piatra Neamț. My parents were teachers, and we lived in a house chock full of books. Looking back, even though I was enrolled in music school, playing violin and doing gymnastics like most Romanian girl of my age, I think I was a bit of a nerd. As I spent a lot of time with books on mathematics, logic, and analytical skills. I knew early on that I want to be a scientist. At first, I thought I would be a mathematician. I was in a prestigious high school, Colegiul Național pentru Rareș. I can't speak Romanian when I'm thinking in English. And most of my friends are mathematicians. And now they're professors all over the world. But as for myself, I loved chemistry, I loved genetics, so I decided to study biochemistry and become a geneticist and molecular biologist. Well, that was a very nice dream for a 17-year-old teenager. Fast forward 40 years, I accomplished all of that, and now I am a geneticist and molecular biologist, but also a neuroscientist. My journey started with biochemistry in Bucharest, the Faculty of Chemistry. There was a Polytechnic Institute, but we were all over the capital. And I had amazing professors who encouraged and even fueled my dreams. Mind you, I graduated in 1989, and dreaming about basic science was quite a wishful thinking activity at the time. But the mentors around me believed in a better future and encouraged us rather than tame our dreams. The biochemistry curriculum was in Bucharest was really heavy on chemistry, but I discovered by myself cellular biology, reading and teaching myself, and immediately immersed in that world. At the graduation, I was drawn towards the Institute of Cell Biology and Pathology in Bucharest, which was the best place with cell biology research in Romania. Now, many scientists have a turning point in their career where they find out what they really want to do. Mine was when I met George Emil Palade, the Romanian-American scientist who was awarded Nobel Prize in Physiology and Medicine for discoveries concerning the functional organization of the cell that were seminal events in the development of modern cell biology. In the early 1990s, Palade came to give a seminar at this institute in Bucharest. So during the lunch with trainees, he made a comment that intrigued me. He said that he considered his work on protein regulation by phosphorylation much more important than the discovery of the ribosomes. From the perspective of a freshly graduate, I was 20-some, that was just an intriguing comment. And I was an inspiring scientist, and the body that brought Palade the Nobel Prize was simply monumental. How could anything be more important than that? What was about the signaling that was so important in biology, I was enthralled, and that was my introduction to cellular signaling, and that moment triggered a fascination with signaling and macromolecular complexes that I still have to this day. To work on signaling and embark in basic research, I applied from Romania to several graduate schools in the US, and I was accepted in some, and I joined Dan Kosman's laboratory in State University of New York in Buffalo. We were focused on signaling, how cells acquire and metabolize iron and copper, which are essential metals for the cell. And Buffalo was, as the fame is, full of knowledge. It was at that time, but it was also close to Niagara Falls. And when we were homesick, we would visit the falls. At the end of the graduate school, we, meaning my husband and I, had 2 PhD degrees, and he's a mechanical engineer, and the dual career problem. But we were also each other's strongest supporter, and this was essential towards pursuing our dreams. His dream job with SIGINT technology brought us to Minneapolis in Minnesota. This is where I started to work on molecular mechanisms that regulate cell-cell communication during development. I was a postdoc in a famous laboratory with Mike O'Connor, and we were working on signaling molecules, among which TGF-beta family of secreted peptides, a family of 33 members in vertebrates that are super important for a wide variety of cellular processes, including tissue growth, differentiation, death, as well as metabolic homeostasis. In my own lab at the NIH, I continued to work on signaling. And we focus on the most exquisite type of cellular signaling and one of the most challenging problems in biology today, and that is the development of a chemical synapse. We know quite a lot about development of the axons, how do they reach to their point, and we know more about diseases that happen when synapses do not work well later in life. But the initial assembly of the synapse remains a really enigmatic place. It's far from worked out, and that's exactly where we placed ourselves. And we focused on glutamatergic synapses, which are the most important excitatory synapses in our own brain. Glutamate is actually critical for the development and function of our brain, and the way that, the fact that you're listening to me now is that because your glutamate synapses are hard at work. Now, insects and crustaceans are using glutamate at their neuromuscular junction, which is the junction between motoneurons and the muscle it innervates. Now, these receptors are super highly conserved, from worms to mammals. We all have them. And in our brain, we know quite a lot about AMPA receptors, about NMDA receptors, but very little about these big clad kainate receptors. These receptors are very elusive, they're very low abundant, and they produce pitiful currents. So measuring them is practically out of questions in vivo. Yet, these receptors are also critical for our learning and memory processes, for regulating our mood for several kinds of information processes in our brain. However, mice that lack all the genes coding for kainate receptors, there are 5 of them in humans too, are alive. They're not too smart. They're very moody. They have behavior defects, but they're still running around. And it became quite a problem to try to understand these receptors using all of the traditional model system. Well, in insects, such as the fruit fly, our model system, these receptors are critical for an essential synapse, the neuromuscular junction, the junction between the motoneuron and the muscle. Without kainate receptors, insects cannot hatch into the larval stage. Think about a butterfly egg that can never produce a caterpillar. So if you have defective receptors, you produce flies that cannot fly. So we have all of a sudden a wide range of phenotypes, allowing us to use this powerful model system, the Drosophila, the fruit fly, to understand these elusive receptors and to discover targets for pharmacological intervention. And because I'm talking so much about neuromuscular junction, let me show you how beautiful these structures are. Here, blue are the muscles. In red are the motor neurons. They come from far away. Think about spine and your toes, the muscle that innervated. These little dots, the green ones, are called synaptic boutons. And this whole field that fills the screen right now is about 1 millimeter wide. And then you find that you get access to it just by opening up a larva. So this is amazing accessibility for everything that you need to do, electrophysiological recordings, optical analysis, and we can really get to understand this synapses. Now, each synaptic bouton, this little green dot I'm showing you here in pink in electron microscopy, it has several individual synapses that look exactly like the ones that we have in our brains. So there is an electron dense region. So these are serial section, electron microscopy serial section. That's an electron dense region where on one side you have these little circles. These are neurotransmitter field synaptic vesicles that will be brought to the cleft and release their content, which is captured by the postsynaptic receptors that are basically channels, open the channels and allow the ion flow to go in and the signal to the neurotransmitter signal to be transmitted. Again, there is a huge similarity in between what a fruit fly has and what we have. Everything is conserved, not only the receptors, but a lot of other proteins that pack these receptors in this, what's called postsynaptic density. Well, and I can, on the presynaptic side, so to make a synapse, this receptor must assemble, and this happens in ER and have to travel to the surface. And then once on the surface of the cell, they have to go to the synapses where they get stabilized, again, by interactions with a huge number of other proteins. The receptors then end up relying on these accessory proteins for function. And the synapses have these amazing properties that they will be tuned up and down depending of the needs. So you can strengthen your synapse or you can weaken them. On the presynaptic side, there is also a lot of regulation happening, and I'm telling you about because we know a lot about this. There are signals. Remember the BMP signals. These are TGF-beta molecules that tell the neuron that the muscle is growing. So grow a bigger synapses because I need more. There are all sorts of signals, autocrine, retrograde. Well, I'm not going to throw too much jargon, but suffice to say that all of these questions, how do the structures, the synaptic structure assemble, how is the presynaptic release, the neurotransmitter release, how is the function of the synapse organized and regulated? What are the homeostatic mechanisms that maintain the synapses in sync with the entire network? These are all fundamental problems in neurobiology, and we tackle them all in my lab. Now, I can't stop myself for not showing you a bit of data, and that's the last piece that I'm going to show you here. And here it is another synaptic boutons with only 5 individual synapses shown from the top or from the side here. This is a reconstitution of this. In blue are the neurotransmitter receptors. These are the receiving end. In green is the scaffold that reaches out to the synaptic vesicles and bring them up to the release side. And in red is actually a whole different complex that shouldn't be there. It's a BMP signaling complex that we're marked here with a particular component. And what we learned is that a selective type of glutamate receptors is responsible for the accumulation of these BMP signaling complexes on the presynaptic side. And they make transsynaptic complexes. And in doing so, these local BMP signaling complexes reinforce and stabilize these postsynaptic receptors. And that happened to be the strong receptor. So in the end, I'm telling you about a positive feedback that reinforces the strong synapses and weakens the inactive ones. Think about use it or lose it. Well, this is the closest molecular description that I know of for that particular phenomena that we all know it happens. Now, reflecting on how this came together, I want to just briefly list some of the advice that I received over the years for many mentors, colleagues, and even my trainees. In science, you need some luck, but harder you work, luckier you get. If you have the courage to go where the research leads you, then you will discover something significant. But we all have to embrace new technologies to stay up up front of the field, because if you don't, well, bad things happen. But you have to do that even if they take you outside your comfort zone. Now, this is a philosophical thought that means a lot to many in our profession. There are many worries, good and bad, in our lives, but we all have to try to build and nurture an environment around us where our worries will be mostly good. And as a scientist, it is absolutely essential to remain personally engaged in experimental work. Now, I'm a tenured scientist, and one of the best possible places to do science, including Basel science, but I'm still a dreamer, and hope that many more could follow a journey just like mine, or even better. And this is a ice sculpture festival in Minnesota, and I'm showing you a dream catcher that I carry with me along my way. Now, it's all about people, the mentors that one has along the way, the colleagues, the collaborators, and the people that I had the privilege to work with, trying to guide them to achieve their own dreams. They come from all over the world. So here I can put together this holiday card with their kids telling you happy holidays to you all. Thank you.
Thank you, Ella. The implications of your groundbreaking work extend far beyond the laboratory. They illuminate pathways to address human conditions such as memory disorders and autism.
And Theo, something very important that probably should have said actually, that it's very important for a very mortal disease, which is amyotrophic lateral sclerosis. And this is also something interesting to make as a research. So congratulations,
yes,
Mihaela.
Congratulations on your unwavering dedication and exceptional expertise, Dr. Serpe. And then you are a powerful advocate for advancing our comprehension of the brain's incredible capabilities. And thank you.
All I'm saying is that it's possible. Dream about it, and it will happen. Yes.
Thank you. Thank you.